Hello, I am Juili Satav from B.Pharm final year P.R.Pote Patil College of Pharmacy. Today I am going to explain you about the treatment of diabetes. Treatment of diabetes is mainly um, based on the severity of a diabetes and based on the type of diabetes. Various classes of anti-diabetic agents and hypoglycemic agents are used to treat diabetes. There are basically three types of diabetes, that is type 1, insulin dependent; type 2, it is insulin independent diabetes mellitus; and type 3, it is a gestational diabetes, which is acquired by a pregnant woman during pregnancy. Then type 1 diabetes treatment includes uptake of carbohydrate fats and to maintain a optimum protein count. Then exercising, which is a very important step. After that, taking insulin externally, as in type 1 diabetes body fails to secrete the optimum concentration of insulin in the body, hence we have to take it externally. Then eating healthy food habits, this is the thing which everyone should acquire to have a healthy life. Then frequent blood sugar monitoring, it is the most important step, as we get a control over the blood sugar level, and we can try various methods to decrease or increase it accordingly. Then, as we have to take insulin externally, there are various types of insulin based on its duration of action. First one is short acting insulin, which we also called as regular insulin. Examples are Humulin R and Novolin R. Then rapid acting insulin, example are Epidra, Humalog and Novolog. These are actually the brand names of the APIs used. Then intermediate acting insulin, example are Novalin N and Humulin N. Then long acting insulin, this includes the example of insulin deg, that is Tresiba, insulin determine, which is also called as Levemir, and insulin gargilin. Then type 2 diabetes treatment includes usage of anti-diabetic agents, insulin, then healthy lifestyle, healthy eating habits, regular exercise, blood sugar monitoring and weight loss. Then anti-diabetic agents, actually type 2 diabetes includes usage of various type of oral hypoglycemic agents and anti-diabetic agents. These are classified into mainly 5 classes. First is sulfonylureas, which includes the examples tolbutamide, chlorpropamide, glipizide, glimepiride. Then biguanides, which includes metformin, fenformin and B.Pharm. Metformin, which is also used as a first line therapy in diabetes. And then megalithinites, repaglinide and netaglimide is an example. Then thiazolidine diones, which includes the example of pioglitazone and rosiglitazone. Then alpha glucuronidase inhibitor, which includes the example of acarbose and voglibose. Mode of action of sulfonylureas, actually sulfonylureas acts via stimulating the insulin secretion from its vesicles. It takes place in beta cells. Then basically sulfonylureas gets bind to its receptor subunit, that is SUR1, sulfonylureas receptor 1, then which stimulates the opening of calcium channel. When calcium enters into the beta cell, it forms a complex with a calmodium ion. Then calcium calmodium complex activates the tyrosine kinase enzyme, which is also responsible for the stimulation and secretion of insulin from its vesicle. Thus, as a result, insulin secretion takes place. Then mode of action of biguanides, actually biguanides are morely a anti-diabetic agent than a hypoglycemic agent, as it normalizes the blood sugar level, and the blood sugar level never falls below its critical level. Metformin is an example of biguanide, and it acts via various mechanisms, as it inhibits the gluconeogenesis, formation of glucose in muscles, intestinal absorption is inhibited, then glucose entry into muscle is increased. Then it also increases the affinity of insulin and glucose receptors. Then mode of action of megalithinites. Actually, this action is more similar to the sulfonylureas one. Megalithinites acts via inhibiting the influx or efflux of potassium channels. This results into the depolarization, which causes the opening of calcium channels. Calcium enters into the cell, and thus it results into release of insulin from its vesicles. Mode of action of thiazolidinediones. It acts via activating PPAR enzyme, that is peroxidase fluorinated activated receptor. This is also responsible for the transcription of various genes, insulin then it directly increases the insulin sensitivity and uptake, and also increases its level in the body. Then mode of action of alpha glucuronidase inhibitor. It's simple and acts via directly inhibiting the alpha glucuronidase enzyme. In figure A, you can see oligosaccharides are converted into monosaccharides via the action of AG, that is alpha glucuronidase, and it directly get absorbed in the small intestine, which increases the postprandial hyperglycemia. In figure B, you can see that our alpha glucuronidase inhibitor is directly inhibiting AG, that is alpha glucuronidase enzyme, which inhibits the transformation of oligosaccharides into monosaccharides. Hence, very small part of it get converted into smaller, simpler, complex, simple material, which is gets absorbed in the small intestine. Hence, due to less absorption, there is no formation of glucose level in there is no formation of high glucose level, and thus it inhibits the postprandial hypoglycemia and decreases the level of insulin. uh, Thank you for listening me. Hope you all get it. And if you have a diabetes, just go